minute now recording. So I'll wait a couple of more minutes because people have lots of other duty, and then typically they join a little bit late or stay a little bit short. I mean, because there are so many other meetings going on in the classroom and so on. But we will have the good exposure. I think through recording Now some people even said, they watching this on for their weekend movie.
Right.
And then some people also said some department actually wanted to move their seminar series and they replace their seminar series with this one. But that was too late because we started this on August 26th, and then their schedule already set.
Yeah.
So they may kind of consider this one as their seminar series for the spring semester, and some department already said to me. And then also I got too many emails from Africa saying I want to watch the recording. So that's why I started recording and releasing that recording. But so far, all of the speakers actually give the permission to release this video. So I'm super grateful for your willingness to share your idea and vision and the presentation, and the synthetic biology community is super excited right now.
I think this is a great idea to engage the global community.
Right. And then thanks to you. I didn't believe at the time, when I invite you or many of people, I didn't expect this huge excitement from the community. But now, oh my God, I overwhelmed right now. But always kind of excited. Because you gave fantastic talk, and then your time, and that's fantastic.
Cool. I also like the idea of putting like a rock star next to emerging scholar. I think that attracts a lot of students, a lot of audience too.
And then interestingly, many coincidences also happen. For example, today I didn't know Sriram, the UIUC alum until I kind of look at carefully. And then you are UIUC right now, so it's kind of coincidence. I don't know how that happened, but many
I
things
did it on purpose.
You did. Oh, you chose it.
Yeah.
Oh, that's great.
I know he worked with Nathan Price. I know.
Right.
He developed the Prom model, and then actually I had a postdoc, Ajit, right? He actually worked with the Sriram.
Yeah.
So you know each other before?
Yes. Yeah. I think I recognize the building too. I think, are you in IGB or something?
Of course. No. RL.
RL.
In my office. Yep.
Yes, nice. Yeah.
So Huimin, you have very nice office.
Yeah. You can actually stop by in the future and see the.
You probably have lots of office or lots of lab. Your institute, right?
Yeah.
You have probably multiple office and multiple lab, and
I do have two offices, yeah, and also 2 labs, yeah.
Okay. That's great.
Yeah, I think the offices in Illinois used to be huge. Like Nathan's office and Huimin's, they're like the older ones, and they're like really large. Whereas the modern buildings have like tiny glass boxes.
And then you also have the big, automation facility. I believe I, watched that video many times.
Yeah. In the Institute for Genomic Biology, yeah.
Right. That, operational, many years, right?
Yeah, quite a few years, yeah.
Right. Okay, so I just want to probably start. If you don't have any question. Okay, so good morning, afternoon, evening, and hola, hello, bonjour, niao, konnichiwa, 안녕하세요. Hello all. Welcome back, and thank you for joining us, the 11th seminar, and I call it UIUC Day because, the speaker from, UIUC, and then also, Huimin Zhao, he's also professor at UIUC. So we had 3-week break due to iGEM Jamboree competition and also my 10-day seminar trip with 8 talks at 4 universities and 3 conferences, including AICHE meeting last week. It was so great to see many colleagues, including SynBYSS people. Actually, we had great time kind of gathering together and then chat and then have dinner and so on. And also, of course, I also met Huimin Zhao, today's speaker at AICHE meeting in person since, I don't know, probably a few years ago, at AICHE meeting. Okay, so it's my tremendous honor to introduce very briefly our pioneer speaker, Dr. Huimin Zhao. I believe, he doesn't need any introduction, but I will just kind of mention something. So he's a professor at UIUC and Stephen Miller Chair, and also director of many research centers, including NSF AI Research Institute for Molecular Synthesis, also IGB theme lead for biosystems design. And of course, he was trained by Professor Frances Arnold at Caltech. Of course, she is the Nobel Prize winner. So he is basically my academic uncle. So without further ado, and Professor Zhao, the virtual pro is all yours. Thank you so much.
Okay, thank you for your nice introduction. Yeah, today I just give a kind of brief overview of my current research program. And so my lab actually is working at the interface of synthetic biology, machine learning, and laboratory automation. So I'm very interested in developing foundational tools for synthetic biology applications, particularly in the form of a biofoundry, which I will talk about in a minute. But also we try to develop new protein engineering tools and genome engineering tools. And then we try to leverage this technology platform for 3 main applications. One is related to drug discovery and development. Particularly, we're interested in developing synthetic biology tools coupled with the laboratory automation to scale up the discovery process for novel bioactive natural products, which could be used as antibiotics or anti-cancer drugs. And in the second application, we try to develop synthetic biology tools for the engineering of microbial cell factory for production of chemicals and materials. And particularly, we're interested in developing genome-scale engineering tools for the rapid engineering of microbial cell factories. And recently, we also tried to incorporate machine learning tools to accelerate the engineering process. And then lastly, we're interested in developing synthetic biology tools for mammalian cells. We're interested in developing genome editing tools for treatment of diseases and also for fundamental research like the chromatin structure and dynamics studies. So a few years ago, my lab actually built one of the first biofoundry in the world called Illinois Biological Foundry for Advanced Biomanufacturing. So we integrated more than 20 instrumentations into a large platform, which consists of many of the instruments that we're familiar with, like a PCR, incubators, a liquid handling system, a centrifuge, etc. And we try to actually break the complex workflows for different applications, such as protein engineering, pathway engineering, or genome engineering into process modules, which will be in turn broken down into unit operations. And these unit operations are more generally applicable, so we could actually program those unit operations to create a custom designed workflow for a specific application. And all the instruments are actually controlled by operating system. And we also have a user interface that we can program all these unit operations. So this actually is the focus of the Bioenergy Center funded by DOE at the University of Illinois. We have a team of 19 PIs from 7 institutions, and we try to develop new capabilities for the biofoundry, particularly the system biology tools to characterize the engineered microorganisms, and also the development of machine learning tools for the engineering of biological systems. And also we try to develop an automated cellular engineering platform, and also like a protein engineering platform. And particularly, we're also interested in developing high throughput screening method, right? Either based on in vivo biosensors or more likely mass spec approaches. And then we try to leverage this synthetic biology platform for the engineering of a number of non-model yeasts for production of different classes of molecules like organic acids and lipid-based chemicals. So this is a major effort actually involving many people. And then more recently, we also launched a new institute funded by NSF, which is one of the 7 first AI research institutes funded by NSF. In this institute, we try to develop new AI tools for the synthesis of molecules and also discovery of new materials. So we're particularly interested in developing AI-enabled synthesis planning tool, and also AI tools for catalyst optimization. The catalysts include enzymes and also chemical catalysts. And then we try to leverage those new tools for synthesis of a few high-value chemicals like artemisinin and also an anti-cancer, antifungal drug. And then we also try to leverage this platform for discovery of new materials. And all these activities that should really build upon the AI tools and also the iBioFab I just mentioned. And in addition, we try to build a similar system for chemical synthesis as well. And this institute consists of 16 PIs. Half of them actually are from computer science department. Half of them are from chemistry and chemical engineering. So this is a major effort, and there will be more activities in the future. Yeah, so that's all my brief overview of my research program, but also a lot of ongoing activities at the UIUC. Okay, that's all.
Wow,
yeah.
The brilliant talk and exciting project. I guess, your lab is doing literally all of the synthetic biology project from microbes to mammalian cell. And I believe the robot and automation system with AI will facilitate the SynBio progress and contribute to the global community. So thank you so much for your excellent work and contribution to the society. And I'm looking forward to seeing more results and papers from your lab. Thank you so much.
Yeah, thanks.
Okay, so now the main speaker of today with a longer introduction. So Sriram, today's the rising star, received his PhD in biophysics from the University of Illinois at Urbana-Champaign, the Huimin Zhao currently located in 2013. And Sriram then worked at Harvard University and MIT as a Harvard Junior Fellow and became an assistant professor at the University of Michigan Ann Arbor in 2017. And actually, he is basically, trained by Jim Collin, of course, the synthetic biology father. And actually, I had great time to meet him last week in person at AICH meeting, discussing, after his, award accepting talk, and that's one of the great time, doing, things in person. So I really enjoyed that. Also, he has developed 8 systems biology algorithms for modeling metabolic and regulatory networks including indigo and magenta and caramel, Prom and Gemini. I love the names of this, model and also some, algorithm. And his research has been published in Cell, Genome Biology, eLife, MBio, and PNAS. So he's the rising star, and he's up for tenure right now. So if somebody kind of watching this, seminar, and then invited to write the letter, I believe you will write a great letter, but please kind of emphasize, he's the superstar and then in this community. So he's also recipient of the Howard Hughes Medical Institute, HHMI, and postdoctoral fellowship and Harvard Junior Fellowship. And of course, the one of a great honor, that is the MIT Technology Review's Top Innovators Under 35, and that's called TR35 Award, and Harvard William Milton Fund Award, and a Distinguished Young Investigator Award from AICG Cobra Society, to name several. So he's the truly rising star, and I really have the great honor to have him today. So Sriram, take it away. Thank you so much.
Thank you so much for the kind introduction, Taesok, and organizing this amazing seminar series. I really enjoyed all the talks so far. So hopefully you can see my slides. Okay. Yep. Thank you so much. Yeah. So as Taesok mentioned, my lab develops computational tools for modeling biological networks. And today I'm going to be sharing with you new methods my lab has developed for engineering metabolic and epigenetic networks. So one of the main goals of synthetic biology is to reprogram cell states. Now, if you are a stem cell biologist, stem cell engineer, and you want to convert a cell type A toward cell type like hepatocyte or muscle cell, then your goal is to epigenetically engineer these cells. And similarly, if you're a metabolic engineer and you want to produce a high-value compound, then your goal is to interconvert low-yield strain to a high-yield strain. In all these cases, we need computational tools to figure out what genes to manipulate among thousands of targets. And there are computational tools that only focus on metabolism or gene regulation. But what my lab's doing is developing new methods that look at the crosstalk between different cellular networks like metabolism, gene regulation, epigenome, so that when we make changes in one part of the cell or network, we can predict ramifications in other parts of the cellular network. So just a brief background, as many of you know, cellular metabolism is highly complex, involving thousands of enzymes, even in simple organisms. And these enzymes are organized into a complex network, and these are highly interconnected in that changes in one part of the network can have far-reaching consequences in a different part of the network. And it's really hard to predict without systems biology and computational tools. Similarly, this metabolic network interacts with an equally complex regulatory network involving transcription factors, post-translational modifications, and non-coding RNAs, which sends environmental stimuli and then turn on or off metabolic genes. So recently, scientists have discovered that this interaction is not just one way, but metabolites, in fact, feedback and regulate many of these transcriptional regulators. And that's going to be one of the major focus of my talk today. So how does metabolism affect gene regulation? So in most eukaryotic cells, humans, yeast, the DNA is organized, is wrapped around proteins called histones, and histones and chemical modifications of histones are the major components of what we call as the epigenome. So epigenome is a major mechanism by which eukaryotic cells regulate transcription and the cell state. And so there are different chemical modifications of these histones that can result in large-scale rewiring of the transcriptional and epigenetics of the cell. So for example, histone oscillation is one common chemical modification of histones, and that results in the release of DNA bound to the histones. And so now we result in active transcription of the genes encoded in the DNA. Similarly, histone methylation is another mechanism for repressing genes, and sometimes it can also activate genes bound to histones. And the key connection with metabolism is that the substrate for histone oscillation is acetyl-CoA, is a key metabolic intermediate. And similarly, histone methylation depends on acetinosyl methionine, which is also a key metabolic intermediate used for nucleotide synthesis, amino acid synthesis. And the removal of histone marks also requires metabolic substrates. For example, histone deacetylases require NAD, and histone demethylases require FAD, which are once again key cofactors in redox metabolism. So what this means is that changes in metabolism or metabolite levels can, they can diffuse into the nucleus and result in large-scale changes in the epigenome and gene regulation of a cell. And so, if you are a stem cell engineer or studying cancer epigenetics, you may want to take into account the metabolic state of the cell, because these metabolites influence the epigenome. And similarly, if you're a metabolic engineer who's focusing on metabolism, some metabolic changes might have adverse consequences on the epigenome, resulting in changes of cell state. And these interactions also play a role in numerous diseases. In cancer, there are mutations in metabolic enzymes that can impact the epigenome. And metabolic disorders like fatty liver disease or obesity results in changes in metabolism, which can impact the epigenome and have consequences even for future generations. So it's really important to study this interplay between metabolism and the epigenome. So you can see from just the simple cartoon here that it's pretty complex, and we need systems biology models to understand this interplay more effectively. So my lab's been using what's called as Genomescale metabolic models, which were originally developed for metabolic engineering applications in microbes. Now we are harnessing the same concepts for studying gene regulation and the interaction between metabolism and the epigenome. So these models have taken account all known biochemical reactions that happen in an organism. For example, the human reconstruction called RECON2 has more than 7,000 metabolic reactions, more than 2,000 metabolites, more than 2,600 metabolic enzymes. And these are also organized into different compartments. So a nucleus is one compartment, mitochondria, peroxisome. And so in the model, the metabolite like glucose in the mitochondria is different from glucose in the nucleus or in the peroxisome. So these models capture the underlying biochemistry with great detail. They also take into account what metabolites are involved in which reactions, and what genes encode what proteins, which catalyze these reactions, how these reactions are organized into pathways, and how these pathways fit in the whole network. So conceptually, these are like, people call them as Google Maps for metabolism. And the way we actually simulate these models is very similar to how we use Google Maps. So I don't want to go into the math behind how we simulate this. So I'm going to explain it through an analogy with Google Maps here. So when you use Google Maps, you provide Google with two inputs. The first input is what I'm going to call an objective, which is your source and destination. Right? You're going to go to Boston to LA or San Francisco. So that's your objective, your starting and ending point. But then if you just give that to Google, there are infinite routes on the Google map that can lead from one place to the other. So you need to provide a second input, which I'm going to call as a constraint. A constraint in this case could be you want to minimize cost, minimize time, or you may want to avoid certain highways or construction sites, or you may want to stop by in Wyoming or Nebraska. So, depending on all these constraints, Google can now provide an optimal solution based on these objectives. So that's the same concept we use for modeling metabolism. So we have an objective where we have a source that are nutrients like glucose or amino acids, and the metabolic network needs to produce biomass components like DNA, lipids, enzyme, proteins for the cell to divide. So that's your objective. You have nutrients, biomass components. And then we have various constraints. We can have thermodynamic constraints that talk about reaction reversibility. We can also have transcriptomic or proteomic constraints that say certain enzymes are expressed or not expressed. So using all these constraints, we can now find an optimal route using what's called genome-scale modeling from certain nutrient state to produce these biomass components. So using this method, you can ask complex questions like what happens when you delete an enzyme or change a nutrient source and how that affects the entire cellular metabolism. So deleting an enzyme is sort of equivalent to closing a highway on the Google Maps. So now the algorithm would now try to look for alternate routes, right? Maybe an alternate pathway that produces the same biomass precursor. If such alternate route doesn't exist, then the model might say this knockout is lethal because the key biomass component cannot be produced. We can also change substrates and see how it results in alternate biochemical routes used by the cell. So we also have complex gene-protein reaction relationships that tell you what genes encode what proteins and which catalyze which reaction. So it's a great way to integrate transcriptomics, proteomics, or metabolomics data and simulate the metabolic state of a cell. So far I mentioned that we have these models for different organisms. What we can also do is create what's called as a context-specific model. So these are models for a specific cell type or even cell state. So we can take the human model and then add transcriptomics or proteomics data for a specific cell type like a liver cell, lung cell, or a stem cell, and then create a cell type specific model using omics data. And this is what we'll be using for the rest of the talk. So with that background, I want to jump in to like a couple of studies we have done using stem cells and cancer cells where we have used these models to understand this metabolic epigenetic crosstalk between in different cell types. So in the first study I'm going to talk about, we use these models to understand how metabolic differences between two key stem cell states called naive stem cells and prime stem cells, how this metabolic difference influences their epigenome and their cell state. So the metabolism of stem cells changes over time, and it greatly impacts their ability to differentiate or divide, or their pluripotency, that is their ability to give rise to different cell states. So even during embryonic development, the metabolic requirements change over time. Initially cells rely more on oxidative phosphorylation, then they switch over to glycolysis, and then they switch back again. So there's a nice, every state has its own unique metabolic requirement. And two cell states I'm going to talk about today are the naive cells. So naive cells are similar to the inner cell mass of the blastocyst stage, and the prime cells are slightly more advanced on the embryonic development than naive cells. But then naive cells can give rise to all cell types of the body, whereas prime cells can't. And so we want to see what is it that's different between these two very similar cell types, and we want to study them from a metabolic perspective. So this was a collaboration with George Staley's lab at Harvard Medical School, and we first did metabolomics to see what are the metabolic differences between these two cell types. And these revealed, metabolomics revealed a lot of differences, but there was no clear pattern when you look at the metabolomics data. Some metabolites in the TCA cycle are going up, some are going down, some nucleotides are increasing, some are decreasing. So it's really hard to learn any patterns just from the metabolomics data. So we then overlaid this metabolomics data onto our metabolic model. And I won't have time to go into the exact methods. It's described in another paper here that I'm citing. So essentially what we do is we take this human metabolic model and have this metabolomics data for different cell states, and then we can create these context-specific models. So in this case, we created a naive stem cell model and a prime stem cell model, and then we can uncover nuanced differences between these two cell states. So to uncover specific reactions that are different, so we did what's called as an in silico gene knockout. So we're starting with just 100 or 200 metabolites, but using our approach, now we can use that to infer the activity of thousands of reactions that are in the model. right? So we're moving from few metabolites to thousands of reaction activity, and then we can figure out which reactions are the ones that drive these metabolic differences. So to do this, we do reaction deletion, and then we identify reactions that are more important in like one cell type, like the naive cells compared to the prime cells. So when we did that, we got a handful of reactions that were best explaining the difference between these two cell states. So in the prime cells, we found that reactions in purine metabolism and folate metabolism were higher or more essential, whereas in naive cells, oxidative phosphorylation and pyrimidine metabolism were more important. Another interesting thing we found was that in the naive cells, folate metabolism, the mitochondria was more important. So we are able to uncover compartment-specific differences, even though we use like bulk metabolomics data. So just to orient you, I know if you're not familiar with the metabolic pathways, so in the naive cells, glucose is being completely broken down. So it goes to pyruvate through glycolysis, and then it goes to the TCA cycle and it gets broken down. Whereas in the naive cells, the model is predicting that glucose is being routed, carbon and glucose being routed towards serine, glycine, folate, and the one carbon pathway. So okay, these were the predictions. We next tested these. So the first prediction was that prime cells were reliant more on folate metabolism. So we used a folate inhibitor called methotrexate, and then we tested this prediction. And when you add more methotrexate and measure cell viability, you can see the prime cells shown in red are more sensitive than naive cells to folate inhibition. And I want to note down these are very similar cell states, and using this model, we're able to uncover these really nuanced differences between them. And the second hypothesis is that the naive cells are more reliant on mitochondrial folate metabolism. So when we add a drug that is specific to mitochondrial folate, now the pattern is flipped, and you can see that the naive cells are now more sensitive than the prime cells. So we also did flux tracing to further validate the model. We did tracing of glucose, glutamine, and serine. I'm just showing data for glucose here. Essentially, what we're doing is we're labeling glucose, and if the model predictions are right, the labeled carbon should go into serine, glycine, folate, methionine in the prime cells, and the labeled carbon should go into TCA cycle in the naive cells. And that's exactly what we saw, showing the heat map here on the left. Red is high, and you can see that in the prime cells, there's higher labeling of serine, methionine, purine nucleotides, just like the model predicted. Whereas in the naive state, there was more labeling of citrate, malate, other TCA cycle metabolites. Just once again confirming what the model told us. So, so the main hypothesis now we want to ask is, so why is the cell doing this, right? So one key hypothesis we had was that this glucose, the redirection towards this one carbon pathway, is being used by the cells to produce S-adenosylmethionine, which can be used for histone methylation. And then we went back to the model, and then we asked, measured the levels of S-adenosylmethionine being produced. And we saw that it was higher in prime versus the naive cells. And then we did virtual knockouts, which confirmed that all these reactions are being utilized, and when they were perturbed, they would affect histone methylation in the prime cells, but not in the naive cells. So we once again tested that prediction. So we blocked S-adenosylmethionine production using another drug called DZA, and then we asked which cell type was more sensitive to this perturbation. And consistent with our model predictions, we saw that the prime cells shown in dark boxes here were more sensitive to SAM synthesis perturbation compared to naive cells. And we looked at all three major histone methylation marks, H3, K9, K27, and K4, and we found the same trend. So what this suggests is that this metabolic rewiring produces S-adenosylmethionine, which is then used for histone methylation, which then results in large-scale epigenetic changes in the prime cells. And this might explain why the naive cells have a different pluripotency, that is, their ability to give rise to different cell types than the prime cells, using this based on this metabolic rewiring. And also, we can also use this approach to benchmark sort of reprogramming strategies. So we can characterize the entire cellular metabolism, right? And then we can say if one cell type is similar or different than the other. So as an example, in another related study, we looked at a reprogramming factor, Lin28, and assessed how perturbation of Lin28 can affect reprogramming strategies. So Lin28 is required for naive cells to become prime cells. And when you delete Lin28, they become naive-like. And so we did the same approach I did earlier. We did metabolomics, built models, and then we compared naive cells based on Lin28 deletion to the wild type cells. And we once again saw very similar differences, where the naive cells had high OXPHOS, high pyrimidine synthesis, and folate metabolism mitochondria. So this suggests that this reprogramming strategy of deleting Lin28 creates naive cells that are very similar to the original naive cell type. And we are able to confirm that from PCA2, where when you delete Lin28, these cells have metabolic state very similar to the original naive cells. So this is once again would help synthetic biology efforts for assessing cell state from a more metabolic perspective. So far I talked about one example where we've looked at how metabolism affects histone methylation and cell state. And for the second part of my talk, I want to talk about how the interactions between metabolism and histone oscillation. So histone oscillation is another major epigenetic mark, and it's highly sensitive to metabolism. As I mentioned before, histone oscillation depends on acetyl-CoA, and its removal depends on NAD. And many studies have found that changes in metabolism affects histone oscillation. But we lack a theoretical model for understanding how or which metabolic changes affect oscillation. So we know it changes, but we don't really know what the relationship is, or can we rationally predict how certain metabolic effects can impact this histone mark. So in a follow-up study, we built a model specific to histone oscillation. So we took the genome scale metabolic models for humans, and then we augmented it for reactions for histone oscillation, reactions that catalyze the addition of acetyl-CoA group to histone and transport of metabolites to the nucleus and out. And so now using this model, now we can track the flux of acetyl-CoA, just like we did for acetylinosyl methionine. We can see what are the maximum levels of acetyl-CoA being produced, or on the minimum levels, and then we can use that to forecast what histone oscillation levels could be based on metabolic changes. So we did a first simple in silico experiment and then compared that with wet lab data. And so what we did was we changed the nutrients the model's experiencing, and then we predicted how much histone oscillation could be produced in these conditions. And so we started with a simple experiment where we removed components from the media. So we removed glucose, glutamine, amino acids, pyruvate, and then saw how that affects histone oscillation. Plus means present, minus means absent. And you can see that the model is able to accurately predict when you remove certain components, how that impacts histone modifications, oscillation. And then we did something more complex where we only added one component at a time. And we found something really striking. So when we grew cells in silico and from wet lab data with just one media component, like just pyruvate, cells actually don't grow, but surprisingly, they had histone oscillation. And from these results, what we predicted was that histone oscillation depends on excess carbon flux that is not being used for biomass production. So what I mean by this is that in conditions, for example, where only pyruvate is present, there's no nitrogen source, so cells can't produce biomass, but there is excess carbon that the cells can sort of store in the nucleus as histone oscillation marks. So by understanding this carbon-nitrogen balance, we can sort of predict the histone oscillation levels in cells. So this was one cell grown in different nutrient conditions. Next thing what we did was we took 14 different cancer cell lines. Each were grown in its own unique media and has its own intrinsic metabolic differences. And then we asked, can we now predict these intrinsic metabolic differences and use that to predict histone oscillation levels in these cells? And so we built context-specific models for each cell, and then we predicted histone oscillation levels, and then we compared that with bulk histone oscillation levels from mass spec studies. So surprisingly, we found a very strong correlation, suggesting that the metabolic state of the cells has a strong effect on their epigenetic histone oscillation levels. And there's a nice linear correlation with cell lines having high oscillation flux were predicted to have high oscillation levels. So next we asked, can we predict genes that would, like influence metabolic genes that will influence histone oscillation? So to do this, instead of changing nutrient levels, we fixed the nutrient levels and then we deleted all the metabolic genes in the model and asked what are the genes that could affect this process of histone oscillation. And then we also measured the impact of these genes on the biomass as a counter screen. So we wanted to find genes that when deleted don't have a significant effect on the growth or biomass, but have an effect on the epigenome. And we found a small set of genes highlighted in red that don't significantly affect the biomass, but affect the oscillation levels. And surprisingly, what we found was that many of these genes were mutated in several cancers and are overexpressed in many cancers, suggesting that these metabolic effects could have a consequence on the epigenome. And then what we also found was that many of these genes had physical interactions with many epigenetic enzymes like histone acylase or deacylase, further confirming the mechanism or behind the model predictions. So just to give you an example of a practical application where we could use these models is, we looked at use these models to forecast if the sensitivity of cells to drugs that disrupt histone acylation. So these are called histone deacylase inhibitors that are being used for treating blood cancers and immune disorders. And they kill cells by increasing oscillation. So we hypothesize that if we can identify metabolites that also increase oscillation, they will synergize with these drugs, right? And similarly, metabolites that would decrease oscillation would antagonize with these drugs. And by knowing a cell's metabolic state, you can sort of predict how sensitive they would be to these drugs. So to do this, we took like one cancer cell line, HeLa cells, and then we grew it in different nutrient conditions like glucose, fructose, succinate. We actually did it for like 50 different conditions. And then, so we're only changing the metabolism. And then we added a histone deacylase drug called vorinostat, which disrupts histone acylation, and we measured its potency in these conditions. So if there was no metabolic dependency, then we should see the same inhibition level across these conditions. But in fact, we saw a very strong metabolic effect. And in fact, conditions that increase oscillation resulted in a higher growth inhibition. This were conditions on the left, whereas metabolites that reduce oscillation resulted in lower potency of this drug. So just by modulating the metabolic environment or the metabolic state of the cell, we can change the potency of this key epigenetic drug. And we also looked at biomass as a control, and biomass do not have a significant correlation. And we've also done like follow-up analysis looking at the cancer cell line encyclopedia. And in that too, when you know the metabolic state of the cells, we can predict their sensitivity to different types of HDAC inhibitors. So confirming there's a very strong connection between metabolism and histone acylation, and we can harness that potentially for engineering or therapeutic applications. So we also applied the same method to several other contexts in follow-up collaborative studies. I want to highlight a couple here. In one recent study, we looked at how fatty acids can result in epigenetic alterations, resulting in potentially turning on of cancer-related genes. So in a collaborative study, we looked at mammary breast cells and that were fed with a high-fat diet. And one hypothesis is that there's a strong connection between metabolic disorders and cancer, and theories that maybe these metabolites have result in adverse metabolic effects that could impact the epigenome, which then could result in cancer or epimutations. And in fact, that's what we saw here, is that using our model, we predicted that adding a specific fatty acid resulted in high oscillation and even more higher bulk methylation levels. And then these metabolic changes and resulting in epigenetic changes turns on many pro-cancer genes, like MAP kinase pathway and NOTCH signaling, which can then result in a pro-cancer phenotype. And so we also did epigenomic studies to confirm that the metabolic-driven changes results in genome-wide chromatin changes. In another study related to aging, we looked at like young and aged mice and their ability to respond to an injury. And in this case, what we found was that aged muscle stem cells from older mice have a different metabolic phenotype than young mice. But then using our model, we were able to identify metabolites, specifically those connected to vitamin A and retinoic acid metabolism, which when added actually turned on many of the markers present in young stem cells that were absent in old stem cells. So just by studying this crosstalk between metabolism and epigenome has numerous applications in aging, cancer, and also another study we found in melanoma too, how a master regulator affects metabolism of melanoma cells, resulting in epigenetic alterations, which turns on survival genes. So these models can help you understand the underlying mechanism behind various diseases, and also uncover potential therapeutic targets. So, so far I've talked about the metabolic epigenome interactions, and for the last few minutes, I want to highlight a few other methods that I've developed. And so far I've talked about how metabolism affects gene regulation, but one of the first methods I developed when I was in Nathan Price's lab at UIUC was called Prom, that predicts the impact of transcription regulation on metabolism. So briefly, what Prom does is that it can predict the effect of transcription factor overexpression or knockout, and how that affects the levels of enzymes and ultimately the whole cellular metabolism. So the way Prom works is that it defines probabilities between transcription factors and their target genes. Like examples shown here, when a transcription factor is turned on, it may result in repression of a certain gene or activation, and we can provide quantitative probabilities for these. And Prom determines these probabilities in an automatic fashion using transcriptomics data. So the inputs for Prom are gene expression data, a metabolic network, and then a gene regulatory network that tells you what transcription factors regulate which target genes. And using that, it can predict, for example, how activation or repression of a regulator can impact the entire cellular metabolism. I won't go into details on this, but briefly what we showed was that this method is automated and can result in large, more accurate models than traditional methods that are done manually. So, for example, we built Prom models for E. coli, which has more than 2,300 metabolic reactions, more than 1,700 regulatory interactions, and used data from 900 microarrays. And we were able to predict growth phenotypes in over 1,800 different conditions. So these conditions involve, like different metabolic substrates or deletion of transcription factors. Then Prom can predict what happens when you turn on or off a transcription factor in a specific metabolic condition. Since our original study, people have used Prom to build models for various organisms, and including organisms like tuberculosis. And so just to give you one more example of how you could use Prom, you can simulate what happens when you change the environmental variables like oxygen, or delete a single regulator or multiple regulators. And Prom was able to accurately predict the growth rate of cells after these perturbations. So more recently, we also looked at post-translational modifications, which are another important mechanism for regulating metabolism. So I talked about epigenetic regulation, transcriptional, and PTMs or post-translational modifications operate at a more shorter timescale. They're involved in more fine-tuned regulation of metabolism. But one big challenge is that we don't really know what enzymes are being regulated by these modifications like phosphorylation or oscillation. And so we developed a hybrid method that uses genome-scale models and machine learning to predict when and which enzyme will be regulated by these modifications in E. coli, yeast, and in mammalian cells. So briefly, it uses genome-scale models, flux balance analysis, and fundamental properties of these enzymes, and then builds like a mechanistic machine learning algorithm that then predicts potential sites of regulation by phosphorylation or oscillation. And these are context specific. Some enzymes may be regulated when there's hypoxia, but they may not be regulated in a normoxic condition. So we can make these more nuanced predictions. And we've been using what's called as a mechanistic AI or transparent AI, where we can actually explain how the predictions are made. So one big challenge with all these models is that these are getting more and more complex, and it's really hard for us to understand or explain why a certain prediction were made. And so we've tried really hard to create transparent methods, and they explain how each prediction was made. So for example, I'm just showing a couple of examples here where the model predicted this enzyme, CAD, or carbamoyl phosphate synthase, was phosphorylated in a certain condition. And these lines tell you the way the algorithm calculated these predictions. So it takes into account different properties, and you can see that the Vmax, or the maximum flux, and growth across conditions were the top properties it was used to differentiate potential options. Like it's giving a lower log odds for oscillation, but high odds for phosphorylation. So by using these properties, it's saying for this enzyme in this context, this is the prediction it made, and this is how we made it. And it can do the same for another enzyme here, pyruvate dehydrogenase, where it was predicted to be oscillated in this case. And so you can see the odds for phosphorylation is going down based on its molecular weight and the flux. And we also have made like interactive plots where you can mouse over and see how predictions are made for each protein in an organism in a given context. So I think I'm out of time here, so I just want to summarize all the methods that I've talked about so far. Now I talked about these genome scale models and how we can now expand them and use that to understand metabolic and epigenetic interactions. And I talked about how the metabolism of different stem cell states influences the histone methylation and their pluripotency. Similarly, I talked about in how we can be knowing the metabolism of cancer cell lines, we can predict potency of epigenetic drugs. And I briefly talked about two other approaches we've developed called Prom for understanding transcriptional regulation of metabolism and post-translation regulation of metabolism, respectively. And my lab is now working towards creating these integrated models that not only take into account transcriptional regulation, but also PTMs, signaling, allostery, and try to create more holistic models of metabolism that enable bioengineering and synthetic biology applications. So with that, I want to thank Taesok again for having me here and organizing this great seminar series, and the people in the lab that have helped me do this, especially Kirk Smith and Fangzhu Shen, who led many of these studies, and various funding sources. And thank you so much for your attention.
Fantastic talk, amazing. I mean, you have lots of great, work, and I'm excited. So I guess I have question, but I have already a question from audience. So I'll start with the question from Fang. So he or she said, thanks for the talk. You showed examples where your model predicted successfully, at least qualitatively, the outcome of experiments. Has it ever happened that your models fail to predict? What do you do in that case?
Yeah, that's a great question. So one advantage of our models is that they're mechanistic in the sense when it fails, you actually know like the reason for failure. We can actually go back and can reveal new mechanistic insights. So in fact, for the Prom algorithm, when we applied that to yeast naively, we developed it in E. coli, we applied that in yeast, and it failed, like miserably. And then what we realized is that in yeast, there's a lot more combinatorial regulation, and we discovered new regulation based on this failure. So we have a follow-up paper from Prom where we have the new approach called Gemini, which I didn't talk about, that identifies cases where the model and the experiments don't agree, and it identifies regulators that could explain the failure. So it would say, this transcription factor, the interaction with this metabolic enzyme is incorrect, and you need to remove that. So because it's mechanistic, we can actually go back and see what reactions to add or remove to help improve the model.
Okay, fantastic. While waiting more question, let me check whether we have. So while waiting question, I will ask question. So actually, when I was traveling for 10 days, I was wondering every time what would be the best or optimum way to go from, for example, RPI, Albany to Washington, D.C.? In other words, by train or airplane, also from Boston University to Harvard Medical School. You probably know what I'm talking about because, you know, Boston Green Line Metro is crazy. I mean, complex. I mean, so confusing. So I kind of, for example, at that time, I'm thinking about by food or walking or versus taking the crazy metro. Actually, I decided at that time, for example, walking for a half hour from BU to have Harvard Medical School to discuss something, I mean, with the George Church lab people, based on, of course, multiple factors, time, and also unbelievably beautiful and warm weather in Boston, and that's really unbelievable. I mean, in November, close to 60 or 70, that's unbelievable. Global warming, I guess. And also, I decided, of course, not necessarily logically based on the time. I'm sure it's time, but more emotionally because I actually walk from BU to Harvard Medical School. I never done that before, even though I lived there for many years, and I enjoy more Boston than ever at the time. So
that's the optimal solution. I was about to say you should walk, but so
yeah, I should walk because I mean there's a beautiful area, and then I can walk. Never found that hidden location. But my question here is, I believe there are so many factors, as you mentioned, when it comes to optimization of metabolic and regulatory networks, including metabolic burdens, resource competition, hidden promiscuous enzymatic function, and so on. So could you comment on the remaining challenges and opportunity in this field?
Oh yeah, sure thing. Yeah. So as you said, the objective of the cell changes over time. Like, for example, like the objective of a stem cell is very different from a liver or a neuron. And one big challenge for the whole field is that when cells are rapidly dividing, we know what it wants. So everything I've showed so far, all the cells are rapidly dividing. Like cancers divide, stem cells divide, and so it's really easy for us to use this model because I know what the objective is. But once you get to like a neuron or liver cell that doesn't want to divide, we don't know what the objective is, and that's been like a huge challenge for this field. And people are we are trying to use like the omics data itself, like transcriptomics or metabolomics, to sort of infer what the objective is, but that's still in a very rudimentary stage. So that's one big reason why you don't see models for terminal cells, and all these models are for dividing bacteria or cancer cells. So I would say that is one of the huge challenges. Another thing is obviously the cell-to-cell variation. We are looking at bulk stem cells or bulk cancer cells, but each individual stem cell might have its own unique metabolic state. So another big challenge is creating these individual cell type single cell models and their crosstalk between them. So I think that's another big challenge in this field.
Okay, fantastic. Thank you so much for your answer. Let me check whether we have any question. Okay, so we have the Fang's question answer, and then I'm checking the chat, and I think we don't have. So this is perfect. So we are on time. So I'll thank you again, and then also thank Huimin. I just left. I mean, he just stay until your talk 1 hour, but he has another meeting. He's a busy guy. So thank him again. So to conclude, so thank all for joining and share staying today again. And thanks to Thanksgiving holiday in the US, of course, we'll again have 2-week break like we did 3-week break last time. So we will meet again on December 2nd, Thursday, the same time. And then we will have Professor Lars Blank from RWTH Aachen University, Germany, and also Professor Tal Danino from Columbia University, actually whom I met in person actually 3 days ago when I visited Columbia University to give a seminar. So this is my great pleasure to have him here. He's another rising star, and he's also upper tenure. So as usual, I mean the follow-up Zoom chat will occur via the link in the chat box. So Sriram, I mean, if you have time, please join us by clicking that on. That will automatically put you to the Zoom meeting, and then we will have some audience kind of wanted to talk with you. So with that, I wanted to thank all of you and then see you soon, basically the December 2nd, Thursday, with the Tal Danino and the Professor the Lars Blank from the Germany. Okay, so thank you, and then this is perfect. I mean, it's on time. It's just see you in a minute. Another Zoom chat.
Bye.
Okay, so just click that one, and then that will ask you to kind of whether leave, and then you just need to say okay, and that is how we doing.
All right.
So you found that link?
Oh yeah.
Okay, so I'll meet you there soon, in a minute. And then Tony, thank you so much. I mean, I missed you a lot, and then finally I met you. And then now you will have another break, of course, Thanksgiving, and then we'll see December 2nd. Tony, are you there? Oh, he probably. Okay, so I will leave myself as well, and I don't believe anything else. Okay.